Okay, good morning, good afternoon, good evening to all of you, and welcome to our webinar. We're sorry for the technical issues we had earlier. We wanted to let you guys join in earlier, but we were having some problems, but now it's all fixed and we're all good. So again, welcome to the Access to Space for All series of webinars on conducting R&D in hypergravity and microgravity. My name is Hazuki Mori of the United Nations Office for Outer Space Affairs. Today, we'll have the second webinar of the series, and we'll dive into life science, and especially biology. So before we begin, I'd like to mention some housekeeping rules. So first, please turn off your cameras and your microphones. We're able to mute you, but we cannot turn off your cameras, so please make sure that you have them off. Second, please use the chat box to ask any questions. We will have a dedicated Q&A session at the end. But if you have any questions, don't wait until then. Just keep shooting them in the chat, and we'll try to collect them at the end. Third, my colleague Wenbin will be active in the chat box of providing you with useful links. And also he will put up a questionnaire link. So please make sure to answer the questionnaire before you leave. It would be really helpful for us. Fourth, please use the hashtag #AccessToSpace4All to help us promote this webinar if you're on social media. We're very active on Twitter, Instagram, Facebook, LinkedIn, at UNUSA. So I just wanted to explain about the webinar schedule. Maybe some of you have joined us already last week, but the main objective of this webinar series is to raise awareness of the many types of R&D done in hypergravity and microgravity, and to trigger interest into this amazing field. So through these webinars, we want to provide theoretical knowledge that can support hands-on opportunities, such as the ones we have under Access to Space for All initiative, but also the many other available opportunities in the world. So the learning outcomes of the webinars are basically the fundamental special characteristics and advantages of the hypergravity and microgravity environment, the overview of what type of research can be done in this environment and its applications, and the different platforms you can use to actually do that. And also, we want you guys to learn how to develop an experiment to be conducted in this environment. So from this week, we have dived into life science R&D. In the morning, we had a session already on Vienna time, and this is the afternoon session for us. So this is our second session of this webinar. It is amazing because we have different speakers from all around the world, which I will introduce after this. Next week, we will dive into physiology, after that pharmacology, and then we will move on to physical science, technology demonstration. And last, we will provide more information about the UN-USA available opportunities, talking more about the hypergravity, microgravity track opportunities. And the last webinar in June, we'll be talking about regional activities. So we've invited space agencies to talk about different microgravity and hypergravity activities that they're doing. Okay, so for the webinars, all the updates will be posted onto our Access to Space for All initiative page. And the webinar recordings and presentations will be posted there too. I'm sorry to say it's a bit confusing. It's not the same page, but we will provide the links to the page, which is the one you can see on the screen, which is called the past webinars. So basically, just go to the Access to Space for All initiative page, and then in the middle, you'll find a link that will take you to this past webinar page, where you can find all the recordings, all the presentations of the past webinars. So today I've already put up the presentations that are going to be done today. The videos, they're not ready, but you can see all the PDFs there. Also, our recordings are on the YouTube. So we have a channel, UN Office for Outer Space Affairs. Go ahead and check that out as well. We have a lot of different webinars that our office is providing. So please, yeah, if you're interested, please make sure to check that out. So this is what our Access to Space for All initiative looks like right now. We have 3 different tracks. Our main focus in this webinar series is the hypergravity and microgravity track. We're providing gradual learning steps from left to right. The left side you can see are on-ground opportunities, and then you can see on the right are on-orbit opportunities. As you can see in the yellow circle, we have one opportunity open for applications right now called Drop Test. This one uses the brim and drop tower to conduct microgravity experiments. It is open until June 30th. So if you are interested in applying for a drop test, please look at our website. There are many documents, many other webinars that could be helpful for you. So if you're interested in drop tests, make sure to check that out. Also, we have 2 more opportunities open. In the satellite development track, you see KiboCube, which is an opportunity provided with JAXA, where we provide a chance to deploy your 1U CubeSat from the International Space Station. This isn't exactly in the hypergravity and microgravity track, but it is definitely the next step for the gradual learning step. So you will have the space, you will have 1U to test any experiments on your payload. So definitely it might be one more step towards the future, but if you're interested in KiboCube, please make sure to check it out. We just had a webinar about the 6th round on Monday, so we have all that content on the website as well. And lastly, on the exploration track, you see ISONScope, which is the provision of telescopes for astronomy. This is actually closing at the end of this week, but you still have time. You still have a few days. So if you're interested, please make sure to check that out. So this morning, we have wonderful speakers, as you can see here. So first, we will begin with our professional talk from Dr. Long, who is from the Institute of Mechanics, Chinese Academy of Sciences. He will give us a 45-minute introduction to biology research and development done in hypergravity and microgravity. After that, we will have 2 student talks giving examples of biology research and development. So the first talk will be from Miguel. He is from the University of Manchester. And then we will have another talk from Silva and Eline, who is from the Belgian Nuclear Research Center. And after that, we will have a Q&A. So I will pick up all the questions that are put in the chat and ask them to the speakers. So please make sure to write in your questions. As I said, you don't have to wait until this time. Keep sending them in, and we'll make sure to answer them at the end. And just for your information for the afternoon session, as you can see here, we have a person from NASA talking for the professional talk. We have Dr. Sharmila. And in that, for the student talk, we also have a student from the University of Texas at Austin, Kyle, speaking for us. So if you're interested, please make sure to check it out. And also, if you can't make it, I know there's time differences and everything. So we will make sure to record it and post it on our website. Okay, so I'd like to give the floor to the first speaker, who is Dr. Long. Please let me introduce you to him in more detail. So Dr. Long is currently serving as the executive committee member of the treasurer of the World Council of Biomechanics. And he is also the director of the International Society of Biotherology, and he is also the director of the World Chinese Biomedical Engineering Association. He is also editor-in-chief of Cellular and Molecular Bioengineering in Asia, Molecular Cellular Biomechanics, and he is part of the editorial board of the Journal of Biotherology. As you can see, he is an amazing speaker, so I will hand the floor over to Dr. Long. Dr. Long, you have the floor.
Thank you. Let me share my screen first. Can you see the screen? Let me do it again. Share the screen. This one, right? So, can everybody see the screen?
Yes, I can see it. Looks perfect.
Okay. Good morning, good afternoon, and good evening, everybody. First of all, thanks, Ms. Moody, for your nice introduction or kind invitation to give me an opportunity to share our ideas with the audience. So my name is Mian Long. I came from the Institute of Mechanics, Chinese Academy of Sciences in Beijing, China. So my research interest is focused on understanding the mechanical force, including gravity, affect cellular responses, especially in immune responses. So the title I'm going to talk today is Mechanobiology of Mammalian Cells under Space Microgravities. So first, I like to introduce what is the space microgravity and the research platform. So as everybody knows that human beings live on Earth, which is a 1G microgravity. We have a 1/3 microgravity on the Mars and 1/6 on moons. So under the zero gravity, there is no buoyant convection, no sedimentation, and also no hydraulic pressures, which means that those secondary processes on Earth become the primary processes in space. So let's look at what's spatial in space. From the point of view of the physics, surface tension will become the dominate in the space. If you look at the liquid droplet, their shape will become a spheric. If you look at the combustion, the space thermal convection will make the frame becomes arc-like shape. From the point of view of the chemistry, the space microgravity induce distinct assembly characteristics. For example, in the space, we can have the bigger sized zeolite, which is more important for the catalysis industry. In life science, the mass transportation is highly changed in the space, which induces the bone loss, as everybody has known. Sorry. So there's a different type of the platform can be used to make such research activities. If you look at the long-term platform, which including the spaceship, space shuttle in several days or several weeks. Also, we will have a space station in space, which can use to apply the experiment on months or years. We also have a short-term research platform. For example, we can have the parabolic flight, in which each parabola persists about 20-some seconds. Also, we can have a solid rocket missions, which provided several tens minutes for the microgravities. Of course, we have the balloon, which can present about several hours microgravity conditions. However, all this research platform has their own limitations. For example, the space mission is high cost, real opportunities with a very limited resources. Also, we can simply copy the studies on the ground into space. We did a new rational design. For the methodology or instrumentations, we cannot just translate the grand method into space. We also need to have the conceptual innovative methods put in the space. For the payroll, we did a very small one or very simple one because we did a very easy to operate instrument payrolls in the space. Of course, we also have several ground-based platforms for microgravity studies. One is a drop tower, which can present several seconds microgravity conditions. However, generally it's a little bit short for the life science study. We can also use a magnetic levitation to suspend the samples in the solution or in the medium, which can mimic the effect of the microgravity. However, this limitation also applied extra coupling effects between the magnetic field with their own biological responses. At a microscopic level, we also have a rotating cell culture system or random positioning machines, which can put a cell in the instrumentation to run them at a specific rotational velocity for a specific time. However, this kind of instrument is still hard to stimulate microgravity in the space, which they also apply new issues such as the stress distribution as mass transport. So let's look at the physiological change in the space. For a different type of the physiological changes. For example, we have the bone loss, we have the muscle atrophy, we have a neurologically abnormality, we also have the immunosuspensions, as well as the cardiovascular deconditionings. In the past decades, there were several countermeasures has been developed to resist such kind of the physiological changes. For example, we can do the exercises such as the running, pulling, and bikings. Such type of the countermeasures are able to slow down the degradation of the skeletal muscle and cardiovascular functions, but still cannot prevent the development of physiological problems caused by weightlessness. Another way is we develop a different type of the medicine or nutrient supply, but the medicines still have some unknown risk. For example, it's hard to tell the different effects to human body between weightlessness and normal gravity. For different type of the nutrient supplies, of course, they can improve or slow down the physiological change, but still cannot prevent such type of the development of the physiological problems. Another way is the artificial gravities, but it may be most effective way, but still limited by the space, resources, and the techniques. Also, it's mainly counted on centrifugation force, rotation radiance, working duration, and as well as the effects of the working mode and body. So if we look at some data from astronauts, they're still kind of diverse. For example, if you look at the bone loss on the right, the bone mass has been decreased at a pelvis. However, it is increased on the head. On the ground, we can also do different type of the test. For example, use a head-down bed test using -3 to 5 degree for the human test. Also, we can suspend the right tails to let the legs free of the mechanical loading. For the in vitro study, I'm going to give two examples. The left one is the cartilage growing in space. So this cartilage in space presents adequate phenotype with the deep cells and collagen fibers, but they don't have the proper mechanical properties. For example, have a lower mechanical strength. At a microscopic level on the right side, if you look at the one mechanical sensitive protein in the cell, microtubules, the tubular solution in space is unable to form the proper biological patterns as that on the ground. So the fundamental issues in space seems like is human adaptivities. It means all species evolved on the Earth must possess a structure function that adapts their living environment. So maybe we can ask different questions. For example, are the physiological changes of the astronauts adaptive or pathological? How to distinguish them? Or can we, or how do we determine if these changes are reversible or irreversible? Is there any threshold, or can this threshold be enhanced to prevent the bad adaptation? So one of the ideas to address this issue is we can ask, can we start to understand this process from a cell? The ideas came from a couple of the reasons. Number one, the key to every biological problems must finally be solved in the cell, as Dr. Wilson said in 19th century. The second, cell pathology is one of the three bases in modern clinical medicine, together with the human anatomy and experimental physiology. So maybe we can focus on the issue of astronauts' physiology at a cellular level. So if we do so, we can further ask, can a cell sense a microgravity? For example, are the single cells or multicellular systems capable of sensing and responding to the change in microgravity? Number two, how do the cells sense the microgravities? Maybe we can ask, is the gravity of a cell is too small to detect a change in the gravitational field directly? The below give an example. For example, a mammalian cell roughly about 1 piconewton weight. But if we compare with the adhesion force between fibroblasts to the glass, it's written about 6 nanonewton per cells. For the contraction force of this cell in collagen lattice, it goes up to 10 to 100 nanonewton per cells. For cell division, the force goes up to 5,000 nanonewtons. So it means that the gravity of the one cell is much smaller than those force we conventionally observed on Earth. The third question we can also ask, from which way does the cell respond to microgravity? It means that does single cell sense gravity directly? It means either intracellularly or indirectly using environmental mediated effects. Since the cell is also mechanically sensitive, it's easy to assume that cell can sense such very, very small force. But however, there's a lot of the environmental change under microgravity. For example, microconvection, hydraulic pressure, and sedimentation changes. So it's also possible the cell sensing the gravity indirectly. So taking this issue together, maybe we can ask the following questions from viewpoint of the cellular level. How do the cells sense the gravitational change? How do cells adapt gravitational change? Or how do we utilize space microgravity resources once we can understand the mechanism at the cellular level? So the third part of this presentation will give you a few examples or case study from my labs. The idea to understand the cell mechanobiological coupling and the microgravity came from the two lines. The left line is sort of the mechanical pathways, It means that the microgravity will change the stress distribution in organisms and also in cells. They also change the mechanical environment around the cells, finally to promote the alteration of the structure function reconstruction or remodeling. The right pathway is sort of the chemical lines. It means that microgravity also changes the mass transportation in organisms or cells, also further change the chemical microenvironment, finally change the cell physiology and cell growth. So the first case study I'm going to present here is use a very simple in vitro study, which we call mechanical remodeling of a mammalian cell under gravity vector. This work came from a very simple worker hypothesis, as shown in the below. Generally, in the lab, we culture cells on the top of the slides, which means that the gravity vector goes down, and the vector normal to this surface goes up, yield 100-degree difference. So we simply think about, can we flip over the slides or erect the slides edge on to change its angle from 180 degree to 0 degree or to 90 degree for the edge-on cases. Using this very simple way, we measure the nuclear sedimentation along the z-axis, which means the longitudinal translocation. At 24 hours, we find that the red one had the higher values, which means that nucleus can settle down much remarkably than other two cases. But if for the longer time, even for the edge-on case, the nucleus can also settle down remarkably along the gravity vectors. Since the nucleus is usually interact with the cytoskeleton, which is mechanically sensitive to hold the nuclear, so we further to understand cytoskeleton remodeling in these cases. For example, we can staining one of the mechanical sensitive protein actin. We find if both in the at short or long duration, the actin expresses is higher than is higher at 100 degrees than both 0 or 90 degrees. Similar case also found for the momentum, which is one of the component for the intermediate filament in the cell. At a shorter time, it's a similar case, higher value at 180. But for the longer time, even for edge-on, it's still very higher expressions. With this kind of things, it nothing to do with the tubing at a shorter duration, but a similar at higher expression at longer durations. Also, if you look at the more detailed structure of the cytoskeleton, we find that most of the cytoskeleton remodeling occurs surrounding the nuclei, even not only for the actin for the left panel, but also for the momentum for the right panel. The middle is a sort of the quantitative data. So the cell nucleus goes down and actin hold it, but finally the cell have to be attached to the surface even for the downwards orientations. So this means that this kind of the remodeling should be associated with the one thing we call the focal adhesion complex, which means that the cell can be anchored onto a glass surface with this kind of a proteins complex. Of course, there's a lot of the proteins in the focal adhesion complex, even over hundreds for the very latest literatures. We pick up two molecules. One is called the vinculin, another one to the penicillin. Finally, we find that there's nothing to do with at a shorter time, but however, for a longer time, the focal adhesion complex goes down at 0 degree. It means a downward orientation, which is consistent with our previous finding for the cytoskeleton remodeling and also nuclear sedimentations. For instance, a simple way we come to the working model to explain how gravity vector can affect the mechanical stability of the cell in the lab. A cell can maintain their mechanical stability using a cytoskeleton network, including actin, microtubule, and momentum, which connected both one side connect to the nucleus, another side connect to the substrate through the focal adhesion complex. We have also done different theoretical modeling and numerical simulation. I don't have time to talk about detail in this aspect here today, but overall, we can see that from different orientation, we have a different mechanical balance. So finally, we come to the conclusion for this on-ground case study is that this process is a gravitational dominant, which because the gravity potential is significantly higher than the random thermal fluctuations. Also, we can integrate differential regulation, which means a mechanical and biological regulation to interpret this type of the phenomena. Also, this type of the things is a integrated effect, which can accumulation of the infinitesimal mechanical terms. Our second ground-based case study is using a conventional instrument of RCS to understand the effects of the stimulate microgravity on factors of the neutrophil-like cells. This comes from the physiological change of astronauts in the space. If you look at the lower right histogram, in the space, total white blood cells number goes up. However, if you look at the PMN neutrophils, it goes up, but for the lymphocytes, it goes down, which means that this kind of the immunosuppressions is also diverse effect. So if we look at the nucleoside recruitment cascade, the nucleoside can be perfused into the blood vessel and undergo a different process, including capture, rolling, slow rolling, fermentation, and transmigrations. So in this process, both nucleoside and neutrophils are highly sensitive to mechanical forces. So the question we can ask, how PMNs and neutrophils and the cells response to microgravity and how the mechanical biological coupling lead to impaired immune functions. Here, simply we can use the cell line HL60 to put into the rotational cell culture system to let them rotate for the at a specific velocity for specific time, we can connect the superlatent after the rotation. We find that several typical cytokines or chemicals, interleukin-6, interleukin-8, and also MCP1 are highly enhanced, which means that under the effect of the stimulated microgravity, these cells exhibit enhanced cytokine secretions. Also, we can also connect the cells after rotation, put into the biomechanical device we call this flow chambers, which they put the flow to enforce the PMN roll over to the substrate, which coated on the molecules. RCCS stimulate rotations. Simply data show that after the rotation, the rolling velocity and adhere cells goes down significantly, which means that under such conditions, the cell cannot recruit effectively, which may lead to the impaired immune functions. Of course, these two previous case study is ground-based studies. Next, we go to the case study on the onboard test. One is from parabolic flight, another from the satellite, recoverable satellite. The first one we have done is a nucleoside response under watering G-force in parabolic flight. This project has been collaborated with my Germany colleagues. The idea is that we try to understand how the water gravity on nucleoside recruitment, especially focus on understand the aggregation in rolling adhesion dynamics, also the cytoskeleton remodeling, as well as the shell flow and chemical regulations. Let me introduce a little bit about the parabolic flight, which the plan can pull up after reach to the 6,000 meters and throughout and pull up after the 40-degree it goes down, which means that the pull up and pull down tolerates hypergravity and throughout undergoes microgravity. The right panel is a hardware we used in the parabolic flights. So we have done twice. One is 2014 and 2017 in Bordeaux in France. Let me show a little bit about this image. The right upper image is a time course of the gravity force change. So the left imaging is a real-time imaging for this neutrophil rolling over the substrate. Of course, there are two pictures on the bottom is illustrate how we do on the plane. So simply give a few data. The left panel is a rooming hardware for this our instrument on the planes. There is a syringe, there is a lens and a cope, and the flow chamber is inside in the box. So the right two panels give a typical data for the rolling speed of the rolling rate of the neutrophil after flow over the substrate. So the data show that the rolling speed of the monocyte cells was moderated accelerate during the micro-G period, the red bar in the middle, which also accompanied by the clear reduction in rolling rate for the right panels. This Dr. Chukaya, Dr. Woolish is our collaborators for this two campaigns. So the final case study examples is use a recoverable satellite, which trying to understand effects of the space microgravity on the endothelial cell and the rat pulmonary stem cells. The idea is came from the one procedures we use in the Chinese recoverable satellites we called SJ-10, which is launched 2016. The scientific issue is focused on how the how the endothelial cell or stem cell is responsive to the real microgravities. The left panel is our hardwares, which including pumps and valves, microscope lens, or even bioreactor, and also the cooling compartment used to control the temperature. Also the fixed compartment to fix the cells and after the missions. So the right up chart illustrate the timelines for the satellite. The bottom the imaging is real-time imaging when the satellite was flying in the space for total 12 days. So once we get the cell from space, we fix the cell, we can test the cell morphology and also other biological responses. For example, if you look at the morphology change in the space, the left two columns demonstrate the cell shapes after 3 days space missions. The right two columns demonstrate a 10 days mission. So if we look at the imaging, we found that space microgravity can lead to the flatter cells with less nucleus and more flaps on the cell membrane if you look at the arrows here and here. Also, we can connect the supernatant to test their energy metabolism. For example, if you look at the glucose concentration in the supernatant, we find that we have the higher values for the glucose concentration in the space at a shorter time, but higher, but lower L-typed lactate in the supernatant at a shorter time. For the longer time at the 10 days mission, the difference seems disappeared. So this data suggests that space microgravity can suppress energy metabolism with lower glucose consumption and lesser lactate productions. Of course, we also look at this cytoskeleton remodeling, which are the mechanical sensitive. For example, even compared to the data for the shorter or longer mission in the space, the actin protein tends to depolymerized in the space, and also the microtubule a little bit. But however, for the momentum, the expresses goes up. So I don't need to spend too much time for the detailed data, but finally we come to the working model for this type of cells, endothelial cells. So the blue color indicates the down-regulated molecules and single-unit molecules, but the red colors demonstrate the up-regulated molecules. Meantime, in the same satellite mission, we also test the cell differentiation for the right. Oh, sorry about that. Right mesenchymal stem cells. Finally, we find that the space microgravity can affect the hypergenic differentiation potential induced more the binucleated cells, like the arrow indicated, or visible plate-like structures, which means it had a more hypotogenic differentiation potential. Also, we check the hepatic specific function typically for the blooming production. Oh, sorry about that. And CYP415 enzymes, which means that longer mission has a higher expression of blooming and also enzyme production. I don't know why, sorry about that. Also, that's a sort of working model. So for the future, we even have a very more good opportunity for the space mission test. Everybody knows that we will have the Chinese space station very soon. Here, the Chinese space station have the one core module for the space medicine and material science study, and experimental module 1 for the life science and astronomy and space environment test, and also experimental module 2 for the microgravity science and astronomy and Earth system science. We also have two modules, one for the Shenzhou manned spaceship connection and also Tiangong cargo spaceship for the transportations. So we have submitted a proposal for first phase 1 mission planned to 2022 or 2023, which is called biomechanics, effects of microgravity on cell growth or cell interaction in liver tissue. Here we're trying to co-culture both hepatocytes and the little cells to understand the correlation between mechanical environment, microgravity, and the liver tissue engineering, especially under microgravity itself, and also the applied shear stress. So for the conclusions, cell mechanobiology under microgravity comes from one idea, is sort of the mechanical biological couplings. We have tried or are developing two research platforms. One is for the theoretical modeling and the numerical simulation. I don't speak too much today. Another is mechanical and biological validations with different approaches on growth on Earth or in the space. We also may need to develop new concepts, methods, or new techniques, especially applicable in space gravity study for life science. Finally, we can develop a new field of cell mechanobiology in space and combine scientific issues with a payload hardware and also integrate ground-based and on-orbit evidence to illustrate how the cell, a mammal cell, can sense and respond to the space microgravity. That's all for my talk today. Thank you very much for your attention. Thanks.
Thank you so much, Dr. Long. Thank you for this informative presentation. I think people were able to learn about the details of biology and also getting into physiology. I think there was a lot of introductions about platforms. Also, the many case studies that Dr. Long has been doing was very interesting to give you more ideas on the things that you can do in microgravity. I really do like the last page when you made the conclusion remarks, giving us ideas, platforms, and pathways for the future endeavors. So I think that was a really great presentation. And I
Thank
saw
you.
that the Chinese space station was mentioned earlier. It's amazing that what can be done in that Chinese space station. As I mentioned earlier, we have projects with CMSA to provide you opportunities at the Chinese space station. So if you can keep that in your head as a goal in the future, I think it would be great. So thank you so much, Dr. Long. It was wonderful.
Thank you.
Okay, now I'd like to dive into the student talks. We have two student talks coming up. So first, I'd like to introduce Miguel Ferreira. He is from the Department of Mechanical Aerospace and Civil Engineering from the School of Engineering, Faculty of Science and Engineering at the University of Manchester. Miguel is a regenerative medicine PhD student. He'll introduce himself later, but to give you a bit more detail, his work is focused on 3D bioprinting and pluripotent stem cells for tissue engineering applications. He has participated in two European Space Agency hands-on programs to study cell behavior. And yeah, I think he'll explain that more in detail for you. So I'll give the floor to you, Miguel.
Yeah, thank you very much. Just sharing my screen now. Can you see everything?
Yeah, I see it in full screen now. Perfect.
Okay. Yeah. Yeah. Thank you very much for the invitation, and thank you all for being here for these talks. My name is Miguel. I'm a PhD student at the University of Manchester in the UK. I'm also a management committee member of the Student European Low Gravity Research Association, and today I will tell you a little bit about some of the work that I've conducted in the field of tissue engineering in ultragravity, and I will have a special focus on the work that I've done as a part of some of the ESA's hands-on programs. So tissue engineering is a scientific discipline that aims to combine cells, biomaterials, and biochemical factors together in order to restore, maintain, improve, or regenerate biological tissues after disease or injury. So in simple terms, tissue engineers are really trying to create biologically living implants with these different components in order to really regenerate living tissues when they're injured or damaged. And one very important aspect of tissue engineering is really combining these components within the right conditions. And as Dr. Long has just introduced very nicely, the mechanical environment is key in order to understand cell behavior and also to modulate what the cells do and how they behave. And this is something that has been explored in the field of tissue engineering, particularly because different cell types of the body are exposed to different types of mechanical environments. So if we think, for example, about the bones, they are exposed to cyclic compression forces. As you walk, the bones bear the load of your body. When you think, for example, about tendons, which are the tissues that connect bones to the muscles, they are exposed to strain, so tension. So as you move, the tendons fibers get stretched. Of course, if they get stretched too much, they can get torn or ruptured, and that leads to injury. But physiologically, they are always exposed to these type of forces. When we think, for example, of endothelial cells, which have also been introduced earlier, which was really good, which are the cells that line up the inner part of the blood vessels, they are exposed to shear stress. So as the blood flows through the veins and the arteries, these cells that are in the inner part of blood vessels, they can really sense these forces. And of course, that leads to different cellular responses. However, in the field of tissue engineering and mechanobiology, there are other types of forces that are starting to be explored now, but haven't really been explored to their full potential. And one of them is, of course, ultragravity. So we know that when humans go to space, there are differences and changes in the different biological systems they have. For example, musculoskeletal system, you have bone loss, muscle atrophy, also cardiovascular system changes. And some of these changes may actually be due to changes at the cellular level and how cells sense gravity and how they behave differently in these conditions. So considering this, me and my team were starting to wonder if we could use ultragravity and particularly hypergravity for tissue engineering applications. So our concept was really to get some cells, expose them to hypergravity, and then understand how they respond to this type of stimulus and see if we could harness these forces as a way to stimulate the cells for the development of new tissue engineering applications. So we chose, so we did two different works. One of them, the first one was with endothelial cells that make up the blood vessels, and the second one was focused on tendons. So these are different cell types that, of course, come from different parts of the body and are exposed to different types of stress, but we decided to look at how they behave in hypergravity. Another question was how to access hypergravity. How could we conduct these experiments? Hypergravity, or increased gravity compared to the gravity on Earth, is not really easy to find. But our team was lucky enough to participate and be selected to the ESA Spin Your Thesis program, which is a program from the European Space Agency that allows university students to access specialized facilities that actually allow you to perform research in hypergravity conditions. And what we did was we went to the Netherlands, to Radstake, and we have access to the large diameter centrifuge, which is this centrifuge here, which is located under this dome here, that basically has 8 meters of diameter and different gondolas, and as it spins, you can have hypergravity exposure inside of the gondolas to your samples. In our case, they were cells. So I have a quick video here of the centrifuge spinning. That basically, as the centrifuge spins, I don't think this is working very well now. So as the centrifuge spins, you can have a range of hypergravity that can go from 1G, which is the gravity of the Earth, all the way up to 20 Gs. And as you see, it's quite impressive. It's a really large centrifuge spinning. But the good thing about this platform is that, particularly for biology experiments, you can they have quite large gondolas, so you can actually house your whole experiment inside of the gondolas. And in our case, we could fit a whole cell culture incubator in there, along with our samples, which were grown in these multi-well plates. So in terms of results, as I mentioned before, we looked at two different cell types. The first experiment was with endothelial cells. We exposed the cells to hypergravity up to 10 G for up to 16 hours. And what we saw was that when we cultured the cells in tissue culture plastic, so on a stiff plastic surface like a petri dish, we didn't really see many differences. But when we conducted an angiogenesis assay, which is this really interesting test to look at how the cells can self-assemble into these blood vessel-like structures, as you can see here, what we saw was that by increasing the gravity level, the cells lost their ability to form these structures. So you can see here our control, it has a really nice, beautiful network of capillary-like structures. And as we increase the gravity level, the cells were not really able to do this. So this was a really interesting result, and we could really see how cells can really sense this different environment and also have a functional differences in their actual behavior. So then a while later, we looked at tendon cells, at tendon-derived cells, and the results were quite different. So after culturing them in hypergravity conditions, we saw some limited levels of cell proliferation, but we saw a tendency towards the upregulation of tenogenic markers. So these are markers of tendon cells in terms of gene expression, and we saw that they were upregulated, so they were expressed higher when the cells were exposed to hypergravity conditions. And this suggested that hypergravity could actually be used to boost the potential of these cells, for example, for a future tissue engineering application. So just before I finish, I would like to leave you with a word of advice, especially if you are a student. I think if you want to get into ultragravity research, particularly in the field of biology or other type of or any other field, I think first step would be to really start learning about the subject, and you can do that, for example, by attending webinars such as this one or other webinars. For example, Celgra has its own webinar series. We have experts from all around the world doing webinars for us, and we've had for the past few years, and they're available in our YouTube channel. Of course, if you're a university student, if you already have some basic knowledge, you can also start reading some scientific publications. Then second step would really be to look for opportunities. So you can apply for educational programs such as the ones from ESA, also the ones from the UN. Celgra actually has is starting to get its own programs. We are going to have a team doing parabolic flights in a glider quite soon. And of course, get in touch with research labs who are working in the field. And finally, I think the most important part is for you to apply. So these opportunities exist and they're out there. And if you are interested, you really need to go for it and apply for them. And so just to finish, I would just like to thank everyone who has been a part of these experiments, particularly the team members of my Spin Your Thesis team. which were really important to be able to conduct these experiments, and also all the partners and all the institutions that together made this happen. And thank you all for your attention.
Thank you so much, Miguel. Thank you for this interesting presentation about tissue engineering in hypergravity. You showed us a really great video of the centrifuge facility in ESA that links to one of the opportunities of the UN. So as Miguel said, start going to webinars, start checking out the information around you, and yeah, start applying. So thank you so much for the advice as well.
Oh, thank you.
Okay, perfect. So the next team is Silva and Eline. They are from the Belgian Nuclear Research Center. So I'll give you some details about them as well. So Silvana is a molecular medicine and oncology, currently conducting her PhD in space immunology at the Belgian Nuclear Research Center. And she is interested in the molecular mechanisms induced by radiation with a special interest in space biology. And Eline obtained her master's in neuroscience in 2018 after she started working on a PhD project investigating effects on spaceflight on the brain and the skin. So they'll both introduce themselves to you, so I'll hand the floor over to them.
Thank you so much for the kind introduction. Is everyone able to see my screen now?
Yeah, looks perfect.
Okay, thank you. So good morning or good afternoon or good evening. I first would like to take a few seconds to thank the organization for this opportunity. And I hope that me and Eline can give you good examples on how to use altered gravity platforms to perform space biology research and hopefully get you motivated to start your own. So I would like to start with giving a general introduction about space environment and space simulation models, and then I will go and give an example on my PhD project, which is focused on the immune system, and then Eline will take over and talk about her PhD project focused on skin, and then we will talk about a special project that we will have that we are conducting and it will take place later this year. So in space, and I would like to thank Dr. Long for the introduction that he already gave, so it makes my job easy. So indeed, in space, we are subject to several conditions. And the first and most obvious one is, of course, altered gravity. So biological systems have evolved under the gravity levels of Earth. And when you go to space, this alteration of the gravity levels will, of course, lead to alterations in our biological systems. And these alterations can be classified as acute or chronic. And as an acute effect, you can have, for example, space motion sickness, which is a form of adaptation of the vestibular system to the new level of gravity. But the exposure, long-term exposure to this altered gravity level can lead to the deterioration of multiple physiological systems, as Dr. Long and also Miguel talked about. Radiation is one of the major concerns and limitations for space exploration, particularly deep space exploration. And the effects of radiation can also be classified as acute or chronic. An acute effect may arise after a solar particle event, which is a high-dose radiation exposure. And the chronic exposure to the galactic cosmic rays, which are not as high radiation dose, but a low dose rate, but continuous, can lead to several alterations and diseases, for example, the development of cataracts or an increased risk of cancer. And astronauts are also subject to another condition, which is an increased level of psychological stress. This is due to the fact that they are in an hostile environment. They are isolated, and the distance from Earth is big. So this leads to an increased release of cortisol by the adrenal glands, which is also an adaptation to this new condition. But the chronic exposure to this high level of cortisol can lead to maladaptations. So what question still is, what is the mechanism behind this alteration? What is the biological reason for this dysfunction? And the question is also, is this a result of the exposure to one of these stressors, for instance, the altered gravity, or do they work in combination synergistically? Or, for example, do one of these stressors act antagonistically in these dysfunctions? So what we are set out to do is to try to answer these questions, unveil the mechanism behind this dysfunction. And once we do that, we might be able to develop countermeasures to reduce or even abrogate this dysfunction. So how can we answer this question? And what is the case for the development of space simulation models? So as also Dr. Long has said, there are limitations to the studies that we can perform in real spaceflight conditions. For example, the payload is a big limitation, so our sample size needs to be small. And also the duration of our studies as a limitation. And it's also impossible or very difficult to isolate the stressors, to be able to understand if the effect comes from one or the combination of all the spaceflight environment. And this leads to the necessity to develop in vitro models, for example, to investigate cellular response to this combined spaceflight environment. This leads me to my PhD project, which is on space immunology, and we are focusing on T cells. So our immune system is composed of several components, which act synergistically to protect us against an external threat. And T cells are responsible for the regulation of this immune response. They also are responsible for killing infected cells. They are key participants in activation of of other immune cells, and they are also key for our memory immunity. In space, there is already been described a condition that is described as a space immune dysfunction. And on T cells, this is seen as a reduced T cell activity, a diminished number of T cells in circulation, but there is also an increased reactivity. So, for instance, your allergies will be exacerbated in space. And there has already been described to be an interaction between microgravity and radiation. So, in order to try to understand the mechanism behind these alterations already described, we are going to perform our project in two separate phases. So first we will use a cell-based approach using UCAS cells, which are T cells that produce cytokine, particularly in high levels like IL-2. And we will expose these activated T cells to the different stressors, altered gravity, ionizing radiation, and stress hormones. And we will do this alone or in combination. And I say here altered gravity because we are interested in trying to find out what is the alterations that we observed in the gravity that we have on the Moon and on Mars. And then we will evaluate how this alters the production of IL-2. And once we set up a trend, we want to use a more complex model, which consists of T cells, but extracted from donors, so from the blood of donors. We will also activate them and subject them to the same stressors in combination or alone. And then we, through the use of several biomolecular assays, we will try to understand how this changes the T cell behavior. And now, so this is my project, and now I will give the floor to Eline, and she will talk about her PhD project.
Yes, thank you very much. So I would like to speak today about part of my PhD project, which is on the effects of spaceflight on the skin. And more specifically, we want to investigate how the spaceflight stresses can affect wound healing in space. Because one of the clinical problems the astronauts report after long duration spaceflight is that they have a delayed wound healing in space. So wound healing is a very complex process, and it consists of multiple phases. And one of very important aspect of wound healing is the migration of the fibroblast towards the side of the wound and start remodeling the skin tissue. And in this video, you can see a monolayer of cells that has been wounded. And sorry, back please, Silvana. Yes, so after these cells have been scratched and wounded, they start migrating again into the scratch and start rebuilding the extracellular matrix. So we want to investigate how the effect of different gravitational levels, doses of ionizing stress, ionizing radiation, as well as psychological stress, can intervene with this very complex process. Next slide, please. Yes. So what we do, we culture primary dermal fibroblasts. These are the major component of the dermal layer of the skin. And they are the, as I said before, the kind of the construction workers of the skin. So that means they migrate towards the wound and start remodeling the skin tissue. We expose them to a mixture of the different space flight stressors of, for instance, microgravity, ionizing radiation, and psychological stress. And afterwards, we evaluate how their wound healing process is affected. Next slide, please. Okay, so after we have scratched and wounded the monocell layer and exposed them to the different spaceflight stressors, we can evaluate the effect of the migration of the spaceflight stresses on the migration. So on the left side, you see the percentage of open wound area after cells have been exposed to microgravity. And you can see in the microgravity environment, the wound area after 24 hours is still larger than after normal gravity exposure. Then if we go to ionizing radiation exposure, we don't see an effect of space relative doses on the migration capacity of these fibroblasts. However, when we expose them to cortisol at different doses, we see a decreasing migration after exposure to the stress hormone. Next slide, please. Okay, so those were that's part of my research, and I would like also to mention the study of the NASA twin study, where one twin went into space for 1 year, while the other twin stayed on Earth. And in this case report, when the twin that stayed 1 year in space returned to Earth, he had very sensitive skin, mostly in gravity-dependent areas. And his skin was so sensitive that in some cases he had to interrupt post-flight activities. So this highlights the importance of developing effective countermeasures to be able to make sure that the astronauts can be exposed to the spaceflight environment for a long period of time, but still have healthy skin once they arrive on the moon or on Mars, for instance. Next slide, please. So what we would like to do in order to start investigating into countermeasure development is we want to also expose our fibroblasts to hypergravity. And we've also been selected by the ESA Academy to take part in the Spin Your Thesis program, in which we will expose the fibroblasts to gravity levels of 15 and 20 G. And after this exposure, we want to investigate if we can counteract the observed delay in migration of the fibroblast after microgravity exposure. So with this study, we want to furthermore also investigate any space-related defects and gravity-dependent processes on the fibroblast migration. So for to conclude, we would can go to the concluding slide. Yeah. So our PhD projects together, they will give more insight into possible interactions of the spaceflight stressors, and as such will improve the risk assessment for deep space exploration. Furthermore, by including different gravitational levels, we hope to reveal altered cellular function related to wound healing, and in that way find ways to counteract the delayed migration and the delayed wound healing we observe in space. So with this, I would like to end the presentation. Of course, all this work cannot be possible with help from other partners, so I would also like to use this as an opportunity to thank all the people and institutes involved in this research. Thank you very much.
Thank you so much, Silvana and Eline. It was amazingly interesting about the T cells, about the wound healing, about so many different cool things that can be done in space. So thank you so much for that informative presentation. I see a lot of questions coming in. Thank you for the crowd for your engagement. We'll dive into the Q&A now. So if you have any more questions, make sure to come send them while we're doing it. Before I go into the Q&A, I'd also like to mention, please answer our questionnaire form before you leave. And also, as my colleague Wenbin wrote, if you have any colleagues, friends, students around you that could be interested in our series, please help us spread the word. Okay, so I'd like to go to the Q&A now. I've seen a lot of technical questions as well, so I hope they can be answered. So the first question was from Baslet to Dr. Long. Okay. Many thanks for your nice and deductive presentation. In represent to simulation models, which is your preferred microgravity model? So you introduced RCCS and RPM, but do you have any preferences? What do you think of partial gravity research? And will you also have a look at it, for example, on the leukocytes?
Okay, thank you very much for questions. Short answer, for both RCCS and RPM, I guess I prefer relatively RPM because it's totally random motions for the cells or cell flasks. For the RCCS, one sense about the cytodeck speeds when you adhere, say, on the microbeads. And the other things is about the cell or the vessel wall effect, because there's two things. It's easy for the beads to collide to the vessel wall. But however, I would like to say all of this is just stimulate the biological effects, because the gravity is body force, which cannot be removed on the Earth. For the second question, yes, I think partial gravity is very important. That's why we are going to do the parabolic fly in the coming June with my German collaborators, which is designed to test the partial gravity effects use our leukocytes. That's my short answer for the question.
Thank you, Dr. Long. I think that was clear. The second question was from Anthony. How does microgravity influence human cell multiplication?
It's a question for me?
Yeah, I'm gonna send it to you, Dr. Long.
Okay, thank you. I guess it's from my opinion, it's cell type specific. Even though we have a lot of data in the literature to propose the cell proliferation may be inhibited, but we also can assume that there's different phenomena observed either on board or the on ground. Even though the onboard data is hard to connect. For our experiment, I guess we don't have time. I don't have the data for the cell proliferation in the space because we just use a video lens to visualize the cell shape. But for the on-ground simulation study, we found that the proliferation is partially prohibited. That's the answer. Hello.
Suki, you are muted.
No. Can you hear me?
I'm sorry. Yeah, I'm sorry. I was muted.
Okay.
Thank you so much. That was a very clear answer. The next question is from Batut. Great talk. Thank you so much. Do you know when the China space station will be operational? So our experiment, Tumors in Space, has been selected 2 years ago by UNOSA to fly on the China space station. So first of all, congratulations to Batut, to Sarah. Sorry, I guess the first name is Sarah. And Dr. Long, do you have more information about the China space station? So when it will be operational? I saw you sharing slides about it, but if you can give us more information.
I don't have too much more information, but what I learned from the public news that the cool module will be launched very soon. After that, the two experiment module will launched in series. So for my rating, what I assume that is will be next year or later, the beginning of the year after next year's. That's what I learned from the public news. Sorry, I cannot get more specific information for that.
No worries. Thank you so much. And Sarah, we'll try to keep you informed as well because we're working with CMSA on that. Okay, the next question is a bunch of very technical questions. I will send them to Dr. Long. Thank you. It's from Rowena. Thank you for the fascinating presentation. She has 3 questions. So the first is, in microgravity, could there be a relationship between increased cranial bone density and increased ICP resulting from upward fluid shifts? So I'll stop with the first question.
Yes, it could be, even though I'm not done anything about the bone for my study, but from my understanding, it could be because the upward flow increase will provide more neutral supplies. So, which means that, which implies could increase the cranial bone density. That's what I understanding. My understanding, yeah.
Thank you. The second question from Rowena is, could the reduced glucose consumption in microgravity lead to an increased risk of type 2 diabetes?
I don't know because we don't have the data for that. I'm not sure if other colleagues focus on the diabetes, but from our data, we just do some kind of phenomenological observation in our work so far.
Do any of the student speakers have anything to add on this about the reduced glucose consumption in microgravity leading to type 2 diabetes? Or if there's anything to add? Okay, I guess not. Moving on to the third question is, I am interested in collaborating on some research about how gravity, microgravity affects the movements of human germ cells, such as sperm and eggs, through the reproductive tract, as it is necessary for successful conception. Do you know of anyone who is interested in working in this area?
I guess answer is yes. There is colleagues in Institute of Urology, Chinese Academy of Sciences, who are focused on the reproductive biology and the embryonic development. Even they have done the experiment in space using some SJ-10 recoverable satellite. So if you are interested, please send me an email. I try to bridge the link between Rowena and the colleagues, Dr. Lei. That's it, Nori. Yes, Nori.
Thank you, Dr. Long. I see in the chat that Sarah has put her contact as well for the reproductive system. Rowena, maybe you can get in touch with Sarah. Okay, another question. This one, I believe, was to our team, the second team for the students. So all things, okay, all these things, do they affect neurological activity? So what he means by all these things is altered gravity, ionizing radiation, and physiological stress. So do these things affect neurological activity? So I'll give the floor to Eline and Silvana.
Yes, thank you. So yeah, that's also part of my research where I'm focusing on the effect of these spaceflight stressors on neuronal cultures. However, yeah, we are currently still looking into this, but there are other studies that show that mostly, for instance, ionizing radiation can have a significant effect on learning ability of rats and behavior of different mammals. So yeah, there are definitely effects of this radiation on neurological activity, as well as stress. It can negatively affect the behavioral output as well. And as of to microgravity, I am currently not aware of any studies that specifically look at neurological activity in microgravity, but I can assume that there could be an effect as well. So all these stressors, they can influence our behavior. And yeah, it would be very interesting also to see how they combine and influence the neurological system.
Thank you, Eline. Actually, I believe the next question is for you as well. So it's also from Rowena. Thank you very much. It was really interesting. Did you look at the cumulative effect of microgravity plus radiation plus hydrocortisone? So she's just interested if this creates a negative synergistic effect on wound healing.
Okay, yeah, thank you for the question. So this is ongoing work, and we are currently looking into how the stresses combine and affect the wound healing and the migration capacity of the fibroblasts. So we're currently working on this. So unfortunately, I cannot say anything about this yet. But in the future, I hope I will.
Thank you so much. When you have the results, we'll have to have you on a webinar again, Eline.
Thank you.
So,
greetings, everyone. It is a pleasure for me to talk to you today about space biology research, one of my favorite topics to discuss. So I wanted to start out by talking to you about how I happened to get into this area of research, because some of you may be looking at in this field. So my undergraduate degree was in biological chemistry, and I worked with the blue-green algae, and I was interested in how this algae influenced the environment around it. My PhD degree was in molecular biology, and there I was using the yeast model system, Saccharomyces cerevisiae, to understand how the RAS oncogene caused changes in cell proliferation. So in other words, I was using the yeast model system to understand the mechanism of causing cancer in the human system. For my PhD degree in graduate school, which was in neurobiology, I used fruit flies to understand how the brain works, how the brain functions. How do neurons talk to each other? How do synapses manage to connect signals from one neuron to another? And so there again, I was using a simple model system, the fruit fly, to understand how the brain functions in humans and more complex organisms. So when I saw a job opening at NASA that was looking for almost these exact areas of expertise, that is how to use simpler model systems to understand how gravity and radiation would affect humans and biological systems in space, I thought to myself, this is a perfect fit, because using these model systems to understand how more complex systems like the humans function in space is very relevant to my areas of interest, because you can't do these experiments with humans naturally. And so using some of these other model systems, including cell culture and so on, becomes very important. And so that's how I ended up working in this area of research. So then for about 22 years, right, I've been working at NASA doing this research, being very interested in space biology. I've done, space flight experiments on the space shuttle, on some of the commercial rockets now, SpaceX and so on, the Russian platforms, Progress and Soyuz, flown experiments on satellites, done experiments on the International Space Station, and so on. Then about a year ago, I decided that I really wanted to learn more about space policy and how that guided the science that we did in space. So I applied for a position where I would be able to help with this process and to learn about the legislative issues behind science and technology in space. And so I applied, went through interviews, and then I started doing this work. And it was a wonderful, wonderful experience. And in addition to knowing now how the science works, I was able to get an insight and help in the process of how the legislative side behind this science worked as well. And so then a few months ago, when I returned to NASA, I applied for and started a position as the program scientist for NASA's space biology program, where I can then help and where I'm responsible for some of the strategic directions for the kind of research that we need to do in space in order to get ready for long duration missions as we get back to the surface of the moon and onto Mars. So on this next slide, then, what I want to show you is the, what are the overall objectives for this space biology program? So, of course, one of the overarching goals of this program is to understand how biological systems respond to the space environment, and to really identify the underlying fundamental mechanisms that are behind the changes, the physiological changes that a biological system experiences in space. We also promote open science. There are these databases for omics data in the GeneLab data system, as well as non-omics data in the Life Sciences Data Archive that can be accessed by people from everywhere globally. And in a way, we can do this kind of science then together as a community by sharing some of the data that we obtain from the spaceflight relevant experiments. The goal for a research program like this is, of course, to do strong, robust science. We also, one of the important objectives for this program also is to understand the fundamental changes using these model systems so that we can support the goal of human health in space, so that we can inform that process and look into, for example, in certain cases, if countermeasures are responsible, then we need to understand those underlying phenomena in order to understand and support human health in space. And of course, as all research done on Earth as well, this research then is also designed to benefit life on Earth. So then the question comes up of what is the importance of studying microgravity or hypergravity? What do we learn from doing this research? And what it's important to understand is that life on Earth has evolved over millions of years in an environment with 1g gravitational force. And so how we rely on this gravity and what this gravitational force does to our biological system is something that's very difficult to understand if, in fact, we don't go to an environment where we don't have that gravitational force. So the best way to understand what gravity does to us and to the biological systems that we have here on Earth is to actually remove it from this environment to see what changes happen, right? So that's one of the reasons for really studying microgravity. And then of course, the obvious reason is the fact that as we go into space, we're going to be in an environment either where there's no gravity, which is what microgravity is, because we are, for example, going around, spinning around Earth or some other planetary body, so that the effective gravity on us is zero, or we're on a planetary surface like the surface of the moon, where we have 1/6 of Earth's gravitational force on us, or on Mars, also with reduced gravity. And so when we're in these environments, we're going to have to understand how the body will respond to those reduced gravity levels. In terms of hypergravity, we're interested in this for a variety of reasons. We undergo hypergravity when a rocket, for example, launches us off the surface of the Earth, and also when we land. So there's increased gravity in those situations. There's also, in a way, increased gravity when you're in space and you're in reduced gravity, and then you come back down to Earth. Essentially, then your body is undergoing increased gravity. And therefore, it's very important for us to understand, there's Earth's 1 gravity, which is what we're used to, and then what is the effect of going to gravity levels above that 1g, which is hypergravity, and below that 1g, which is microgravity. And so all of this becomes important for us to study and understand in order for us to be safely in long duration missions on the moon and Mars. So this gravity dose response, we do understand that a change in gravity will cause change in gene expression. It will cause changes at the cellular systems and even at the organ system level. But we've seen that, and I'll show you examples later on in this talk. But the dose responsiveness of this gravitational effect on the biology is not fully characterized. So we don't really understand, for example, after we're moved to this altered gravity environment, does it take us a while to adapt? And then after we've adapted, we can resume life as normal at these gravity levels? Or does this, the changes that we experience, do those persist long durations over time? Or, for example, if we are looking at the reduced gravity environment, can we introduce a 1G gravity for short periods of time? And is that sufficient to recalibrate the body back to normal function? These are all things that we need to understand in greater detail. So in order to do that, we have, luckily, at our disposal, both on the surface of the Earth and in space, different research platforms and tools that we can use to do these research topics and understand the science better. So the first example I was going to give you was the International Space Station. So this is, as many of you probably know, it's a platform. It's actually a science laboratory that is in space, that is spinning around the Earth at 400 kilometers above the surface of the Earth, in this microgravity environment, allowing us to do science, to ask some of these questions that I just brought up earlier in my talk. Not only that, as I mentioned, it's a science laboratory, so it has a lot of equipment that allows us, for example, to grow plants in space. You can see over here in the middle that you can grow plants. You can do cell culture experiments in space. You can have animals, vertebrate and invertebrate animals that you expose to this environment in space. And in terms of this hardware, for example, something I wanted to point out to you is there are some habitats like this one that have a centrifuge built into them. So what you can do when you do your science is you can have one set of samples that is in a 1G centrifuge in space, along with your samples that are in microgravity in space. So in other words, those two sample sets are experiencing everything identically, except one set is in microgravity, the other set is in space in 1G, but both are being exposed to radiation and any other conditions that are different in space versus ground. And then you can always have an identical third set that is on the ground and under Earth's 1G environment, and then compare these three data sets, or at least the two data sets in space between microgravity and 1G, to look at the effect of gravity in the space environment. And then on the top right here, what you see is a glove box. So this is to give you a sense that you can do experiments in space somewhat like you would do an experiment on Earth in your laboratory, where if you needed to fix a sample or use certain chemicals that you don't want to spread around around the room, then you can use a biosafety cabinet, or you can use a hood, as it were. So you have that sort of a capability also on the International Space Station, and you can use those facilities to do your science. Then there are other examples of other habitats that you can use, for example, the rodent habitat. And a little bit later today, I'm going to talk to you about rodent experiments, mouse experiments that help us learn about some of these changes happening in the space environment. On the left here, you see the Japanese Space Agency has a mouse habitat with a centrifuge in it. And we just talked about the value of having a centrifuge in space so that you can control for the gravity vector, as opposed to all the other changes that you could experience in space. You have instruments like the DEXA scan, where you can look at bone density. And one of the things we know is that when you're in the spaceflight environment and your body is not loaded with gravity the way it is on Earth, that you could have a reduction in bone density. So you could lose some mass in the bone, as well as in the muscle, for example, which could weaken some of these tissues. And so you can do studies for many, to ask many of these questions in space. In addition to the International Space Station, there are other venues, other platforms that you can use to look at partial and reduced gravity environments. For example, there are suborbital flights or parabolic flights, for example, which will expose your samples to short durations, a few seconds or a few minutes of the microgravity environment. And you could test your instruments and your science in that way. There are the Russian platforms like the Bion missions where you can look at microgravity environment as well. And then coming online, which will allow us to do a lot of science on the surface of the moon, as well as around the moon and beyond, are these other vehicles that I show you at the bottom half of the slide. On the right, the CLIPS, or the Commercial Lunar Payload Services, will take science experiments to the surface of the moon so that we can look at what the changes are in this environment of partial gravity and increased radiation. Similarly, the Lunar Gateway, which is the station that will be orbiting the moon's surface, one can conceive of putting small experiments, or small payloads, as we call them, into that environment to look at the changes that would result on the biology. Similarly, on the Artemis missions, you can, these are the Space Launch System rocket that's going to take humans to the environment of the moon and beyond. You can start to send up experiments in vehicles such as this to understand the effect of the space environment. And then I wanted to give you a few more ideas of some other tools that we can use on the surface of the Earth, for example. So ways in which we can simulate hypergravity or microgravity on Earth. So for us in the space biology program for NASA, we use two centers, the Ames Research Center, which is in California, and the Kennedy Space Center, which is in Florida. And we utilize the facilities that they have to do some of the science. So this first example at Kennedy Space Center, they have the microgravity simulation support facility. And to give you an example of what I mean by this is, so we were talking about doing science in space earlier, right, on the International Space Station or on these rockets and taking them to space. But you can imagine that those opportunities come few and far between. They're expensive. And so another way in which you can start doing experiments so that you can repeat them in your lab, get comfortable, test them, and make sure that they're working fine, is to use these platforms to simulate microgravity on Earth. And so one of the examples shown over here in panel D is what we call the rotating wall vessel. So here what it is you can have cells or C. elegans or worms or tardigrades or other model systems which can be suspended in a fluid, and then the apparatus actually rotates so that the cell or the model system that you're studying is in a constant state of free fall. And so in a way, you're simulating the effect of weightlessness, although it is not truly in a weightless environment. And you can do this in the lab, and you have access to your other equipment. You can repeat your experiments as you need to. And so it's one way to get your experiments tested and ready for a spaceflight mission. There are other tools like the random positioning machine, for example, which can randomize the gravity vector by changing the direction constantly, randomizing the direction of the gravity vector so that the net force on the cell or the system that you're exposing to this facility is randomized so that there's no net gravitational force in one direction the way you normally have it on Earth. And as you can imagine, these are simulated microgravity environments. They're not true microgravity, but you can use them on Earth in laboratories to start to see what the effects might be. Similarly, then, there are hypergravity facilities. We talked about hypergravity being another force. And so at Ames Research Center in California, for example, there are these centrifuges where you can load samples which are, plant samples or animal samples, which are then exposed to gravitational levels above 1G. So you can look at the effects of hypergravity on your sample. On this slide, what you see are, I've talked to you about some ways in which we can use platforms on the surface of the Earth as well as in space to do the research so that we understand, how space biology functions. I wasn't able to necessarily describe every option, but they're here as a reference for you if you're interested. One thing I did want to point to you is down here on the left side, where you see the picture of the mouse. I wanted to talk a little bit about rodent unloading, because I'm going to talk to you about this later when I give you examples, research examples. So another way to simulate weightlessness or partial weightlessness for animal models like mice or rodents and so on is to use a harness and then lift the animal, so-called partial loading, or even lift just the hind limb of the animals, which is called hind limb unloading. So do partial unloading of the animal to look at the biological effects of those changes on the animal system. And so, and we'll talk about that as another way to simulate microgravity on the Earth's surface. So now I wanted to switch to specific research examples so that, and these are, most of them are published works which are out there in peer-reviewed journals that have come out quite recently in the last few years. And so I wanted to use those examples to show you the kind of research that is happening right now and what we're learning from them. So in this particular example, what this paper talks about is, this is a rodent experiment. So mice were exposed to the spaceflight environment on the International Space Station for 35 days. And then those samples were used to look at the brain. They were used to look at tissue integrity and tissue morphology in the brain. For example, looking at the vascular endothelium tissue, looking also at astrocytes and glial cells, looking at proteins like tight junction proteins, which are important for membrane integrity, to see how these changed in the spaceflight environment. And what they found was that these components, the proteins, these particular cells that were important for membrane and tissue integrity were altered from animals that were exposed to space, such that these changes could actually result in tissue integrity, which could change important brain functions, like, for example, the blood-brain barrier function. As you know, the blood-brain barrier is a very important brain function that separates the brain, protects the brain from toxins or pathogens, microbes, from coming into the brain and affecting brain function. And so you want that barrier, that a tissue barrier, to be strong. And so when spaceflight affects cellular function and protein function and the integrity of and remodels tissues, then it becomes important to study because you want to understand how it might potentially alter the function of the brain in this case, for example. Now, this next study, this next publication, is actually a fruit fly experiment. So that first one that I showed you just now earlier was using the mouse and looking at the brain. Now this study uses the fruit fly, Drosophila melanogaster, to look at the cardiovascular system, to look at the heart. And what you see here on the left side is, and marked in red, are these actin filaments. These are myofibrils. So these are circumferentially arranged in the Drosophila heart. And so on the left side, what you see is that these myofibrils, when they contract, they allow blood to flow smoothly through the heart tube. On the right side, on the other hand, is the heart from an animal that has developed entirely in space. Now, here's an advantage of using these model systems like invertebrates, which have a quick generation time. And so on a 30-day mission, you can get, 1 or 2 generations of flies that have entirely developed in space. And on that right side, when you look at the hearts, you'll see that those red myofibrils in the middle are very disorganized compared to the myofibrils on the right. Similarly, in green, what you see is the membrane, the extracellular matrix that sort of binds and is a structural component that stabilizes and attaches to the myofibrils, and that is highly disorganized as well. So in other words, what you're seeing here in a very different model system, you're seeing also that there's tissue remodeling from animals that have developed in space. And in fact, in this particular case with the heart, these changes actually cause heart dysfunction that have been measured in these animals. And there are also changes in the proteins, the proteostatic stress that is caused in the system is also seen, similar to the fact that there are alterations in the protein signatures also in the brain in the mice. So 2 very different systems, 2 different organ systems, 2 different model organisms, and yet you're finding in both cases that there is a reorganization or remodeling of tissues that are important for function. Next, this particular study looks at biomarkers. So one way in which you can really measure the status of a human or a sample that you sent in space is to have a biomarker that you can quantitatively measure. So this paper reports looking at the blood cell counts for white blood cells. And this study looks at the leukocyte counts in astronauts that have been exposed to the space environment, to rodents, to mice that have been exposed to the space environment in space, and also to those cells and animals, mice that have been exposed to simulated microgravity on the surface of the Earth. And in all of these cases, it was found that there seems to be a common change in the ratio, an increase in the ratio of the neutrophils to the lymphocyte. So the NLR, the neutrophil to lymphocyte ratio, seems to increase in all of these cases where the biological system, i.e. the human or the mouse, is exposed to this spaceflight environment. And so this paper reports looking for measurable biomarkers to assess health status for humans and animals in space. Now, one of the things that I wanted to talk to you about is, so far we've been discussing gravity. We've talked about microgravity or zero gravity. We've talked about hypergravity and increased gravity. But what I also wanted to talk to you about is radiation. So as we go into space, and in the bottom left over here, you see a pictorial, this little blue marble is us on Earth, and we are surrounded by a magnetic field that actually protects us from some of the harmful charged particle, highly damaging radiation that comes to us in the form of galactic cosmic radiation. There's a background of this galactic cosmic radiation, particles like iron, charged particles like iron, carbon, oxygen, silicon. These are particles that are constantly going to bombard our biological systems as we go into deep space beyond this protective magnetosphere that surrounds us on the surface of the Earth. So even the International Space Station is protected by this magnetic shell, and so we call that low Earth orbit. And the minute we go beyond this, beyond 58,000 kilometers, beyond the Van Allen belts, then you experience what is called deep space, where you have these elevated radiation levels. And so far, the limits of our understanding is long durations in low Earth orbit on the International Space Station, or short duration missions during the Apollo years to the moon and on the surface of the moon. But we now need to understand what those longer durations in the deep space environment with this combination of radiation and reduced gravity. And so I wanted to give you some examples of this sort of research. And this first one, what it shows you is a study looking at vascular dysfunction. So looking at the function of a microvessel in the hind limb of a mouse. And in this particular case, the mouse is exposed to simulated microgravity by doing that hind limb unloading that I told you about. So this is a way to simulate weightlessness by artificially unloading the hind limb of the animals, and also exposing the animals to radiation. So exposing it to this iron, which is a component of galactic cosmic radiation, and to expose this, and even though 1 gray, which is a pretty high dose of this particular radiation type, but what it shows, interestingly, in the study, and what you see here in the graph in the middle on the lowest curve, is that the biggest detriment to the function of this blood vessel in terms of vasodilation, the biggest decrement is seen when the animal is exposed both to radiation and to simulated microgravity. So when you have the combination of the stressors of reduced gravity and radiation, you see the biggest effect in terms of loss of function or dysfunction of the ability of this vessel to dilate the way it should normally do. And this paper also talks about the fact that the signaling pathway nitric oxide seems to play a role in mediating this particular change. The second example is another example using mice and looking at the brain this time, right? And in the brain, in this particular case, this study used a different kind of radiation, gamma radiation, which is ionizing radiation, but is different from the kind of radiation that an organism would experience in deep space. So it's different from the galactic cosmic radiation particles or solar particle events. But what is also relevant, I mean, it's ionizing radiation, but it's also relevant that it's a low dose rate. So using a cobalt plate, this group was able to expose mice to this low dose rate of radiation, which is similar to the kind of radiation that you would experience in space in terms of the doses. And what this study found is that here also, if they simulated weightlessness with hind limb unloading and they exposed the animal to this low dose rate of gamma radiation, even 9 months after, after you exposed it to this combination of radiation and simulated microgravity, even 9 months later, when you looked at the brain, there were increases, significant increases in oxidative damage markers. For example, if you looked at lipid peroxidation markers, there was a significant increase, and the biggest decrement, the biggest increase of this damaging marker was found in the case where the animal was exposed both to radiation and to simulated microgravity. So here are examples where, again, it's very clear that the effect of exposing the animal to two different stressors, which we know will be present as we go into deep space, the presence of both these stressors causes the biggest change in function of the biological system. So that's an important thing for us to keep in mind. Now, this paper, I wanted to talk about astronaut study. This is an older paper from 2008. And what this one shows is that when, and this was all done in low Earth orbit. So when astronauts, either on the International Space Station or on the Russian Mir station or on shuttle missions, were exposed to roughly equivalent levels of radiation. When you compare the blood from these astronauts before the mission and after the mission, and you took these lymphocytes, the white blood cells, and you looked at the chromosomes, and you looked to see, you actually quantified the frequency of aberrations seen in the chromosome, it was found that there were significant increases in the frequency of these chromosome aberrations in the lymphocytes, in the white blood cells of astronauts that had been exposed to space compared to the controls that had not been in space, but also more importantly, because different people can have different responses. So more importantly, the comparison was for the same astronaut cohort before flight and after flight, there was a significant increase in the frequency of these chromosomal aberrations after flight. And then in this other paper from 2017, what they talk about is when you look at this lower curve with the dark circles, the filled circles, what that shows you is the plot of increasing amount of damage, so increased chromosomal aberrations in terms of translocations in lymphocytes as you increase the number of days in space. This is in low Earth orbit, this lower curve. So that is the International Space Station. Now, remember, I told you as you go beyond the Van Allen belts, you now have increased exposure to radiation. And so this upper curve here with the empty circles, what that shows you is the projection of what we anticipate to be the frequency of these translocations in the deep space environment, with the increased amount of radiation. So for the same amount of time spent in deep space for an astronaut, you anticipate getting a much larger change or increase in frequency of this damage, this radiation-related damage that you can measure in terms of chromosomal translocations. You expect to see a much larger increase in that damage in the deep space environment. So that's also important for us to keep in mind, and further strengthens the need for us to do this kind of research. And the last example that I was actually going to show you is relates to what I just talked to you about in the previous example. But the difference is this study was done in a test tube. So the previous study was using astronauts and taking blood from them before flight and after flight. This particular study looks at the same assay. So it looks at the chromosome, looks at aberrations. You can paint your chromosomes, as you see here on the right side in the pictures. You can paint the chromosomes with a fluorescent tag so that you can look to see after exposure to radiation or after exposure to simulated microgravity, you can measure and quantify the extent of chromosomal aberrations. And what you see over here is, in this particular case, is on the left side. So these are using the human blood lymphocytes, but you are using that in vitro in a test tube. And what you see on the left side here is that when you expose the human lymphocytes to simulated microgravity, and this is, you're using that simulated microgravity, the rotating wall vessel that I told you about, and you're exposing them to radiation. In this particular case, the carbon particle, or the carbon ion, which is similar to the galactic cosmic radiation particles. The left side is a control. So what you see here on the left side is that as your carbon ion, as you increase the dose of radiation, you will, of course, see a loss in viability of the cell. So that's the control. In other words, the more you irradiate the sample, the more there are dead cells in your test tube. And that makes perfect sense. And what this shows you is the fact that carbon ions, this kind of particle that will be in galactic cosmic radiation, is more damaging than something like X-ray, which is a different form of radiation that we know about from Earth, from cancer therapy, for example. And why I call this a control is because on the left side, you see that regardless of whether you expose the cells to both simulated microgravity in the rotating wall vessel and to radiation, or you don't do that, either way, the cells show the dominant effect here in loss of viability is the exposure to radiation. The simulated microgravity has no effect on cell viability with increased dose of radiation. So that's a control to say that this system works as we would predict. On the right side is an interesting data set, and what this is showing you is this chromosomal aberration frequency that we were talking about. And what you see here is that, as you would expect, the carbon ions cause more damage. So for the same dose of radiation, the carbon particles cause more damage than the X-ray over here, which again is something you would expect. But what's very interesting is that in this particular case, what you see is that if you have both the exposure to the carbon radiation and you have simulated microgravity using this rotating wall vessel on Earth to simulate the lack of gravity, you see that the biggest effect, the biggest change or increase in frequency of chromosomal damage is seen in the situation where you have both the carbon ion and simulated microgravity impacting the system. So there again is an example. And this is an interesting study because this uses a cell culture system in a test tube to start to parse out the effect of gravity versus radiation and quantify the differences of these environmental effects. And then here, to end, I wanted to summarize and tell you then that what I talked to you today about is that space biology contributes to basic research. It helps in the expansion of knowledge, so we understand the effect of this environment, and also to really start to define the fundamental mechanism of the changes that a biological system will experience when it is in space. And to do this in space biology, we use multiple model organisms spanning from the simplest unicellular systems to complex multicellular plants and animals to understand, how the spaceflight environment is going to affect biology. Another thing we talked about is that in spaceflight, organisms will experience multiple stressors. We talked about reduced gravity, microgravity, partial gravity, and hypergravity. But we also talked about elevated radiation. And there are other environmental stressors in space, for example, increased carbon dioxide levels, for example, a change in circadian cycles. On Earth, we have the sun, 12 hours of day, 12 hours of night. Some of those cues are missing in space. And so it is important for us to understand the effects of all of these stressors in the spaceflight environment so that we can develop effective countermeasures if we need them for long duration exploration of humans to the moon and Mars. And with that, I thank you for your attention, and also thank you to Dr. Anthony Hickey, who has helped me put some of this slides together for this talk. And with that, thank you very much for your attention, and good luck with your research career as well. Okay.
Okay. I'd like to thank again Dr. Sharmila. She is not present in this webinar right now, but she apologizes for not being here. And if you have any questions to her, please, you can write it in the chat as well. Some of them we might be able to answer, but others, please leave her a comment in the questionnaire form. So there's a comment section. So if you have any questions, please write it there and we will share it with her. So Dr. Sharmila is the program scientist of space biology in the Biological and Physical Science Division at NASA headquarters. And as you heard, she's been working on space policy as well. So if you have any questions on space policy, maybe you can ask her as well. Okay, I'd like to move on to our next speaker, who will give us an example of biology research that is being done in hypergravity and microgravity. So please let me introduce to you Kyle. Kyle Morgenstein. Sorry, Kyle is an experimental astrobiologist researching the preservation of microbes in deep time. At MIT and with NASA JPL, Kyle studied the preservation of proterozoic cyanobacteria. He's going to explain it to you guys in detail more, but he's been doing that and how their preservation may inform the search of life on Mars. So he has an exciting presentation for us. I will give the floor to Kyle.
Thank you so much for that introduction. Let me share my slides real quick. All right.
And if you have any questions, Kyle will try to answer them as much as possible, including the ones to Sharmila. So he has a big responsibility in our webinar. But anyway, if you have any questions, please keep writing them in the chat. Okay, Kyle.
All right. Can you see my screen?
Yeah, looks good.
All right, great. Let's get started. Thank you so much for that introduction, and thank you for having me. My name is Kyle Morgenstein, and I am an astrobiologist. And we'll talk about what that means in a moment, but it allows me to wear a lot of different hats. So in my past, I've been able to do geology research and microbiology research and now robotics research. And all of these things together help us ask questions about the nature and origin and evolution of life and its proliferation both here on Earth and maybe its proliferation throughout the universe. And this allows us to ask questions from a variety of different fields. There's, of course, the basic physics and chemistry that underpin biological systems, but it's also a philosophical question. When we think about finding life on Earth, we have a good operational definition for what that means. We're looking for certain metabolisms and transfer of energy and reproduction and response to environmental stimuli. But on other planets or other parts of the universe, life might not look the same. It might not even be carbon-based. And so understanding what it means when we say, is there life here, almost becomes a philosophical question. But today we're going to take it out from pie in the sky land and bring it down to Earth, literally, and we're going to focus mostly on this geology bubble. And so the questions that we're going to really think about are, what evidence do we have for how life evolved here on Earth? And then how we can use that evidence to inform the search for life elsewhere in the universe. And in doing so, we use Earth as an analog. It's the only data point we have for life existing anywhere in the universe. And so we use it as a model to understand what we look for when we travel to places like Mars. And then we're also going to touch on how we go about collecting this geologic data. And so we'll get to robotics in a little bit later here. So first, let's think about what I mean when I say geologic evidence and how that helps paint a picture of past life. When I talk about past life, a lot of people think of things like this, a dinosaur skeleton. And this skeleton certainly paints a picture of past life on Earth. Something like a skeleton can tell us about the diet of the organism or how it walked or its reproduction or the type of environment it lived in. But we also have to understand its limitations. For example, maybe the Triceratops had a lovely singing voice, but this fossil can't tell us that. So we have to try and find data that is specific to the types of questions we're asking. However, there's some big problems. For one, evidence like this fossil has only existed for about the past 540 million years or so, since the Cambrian explosion. Prior to that point in Earth history, we had no animals or plants that were capable of being preserved in a fossilized way. They had no hard structures that were preserved in the rock record. But we do know that there was life much, much farther back in Earth history. Earth is about 4.5 billion years old, and the oldest organisms date back to maybe 4 billion years. So that's the majority of the history of life on Earth. So only about 10% of that history do we have any kind of hard fossil evidence of those life forms. but we know that life was doing quite a bit even before we got to this animal and plant stage. For example, 2.2 billion years ago, we had the evolution of photosynthesis, and that's when you start to see oxygen appearing in the atmosphere and the ocean. And we know this through a variety of different means. But what we want to understand today is what kind of geologic evidence can we gather about this past life, and how can that inform the search for past life on Mars, where we don't expect to find a full T-Rex skeleton just sitting on the surface waiting for us, when we expect to find life forms more like these microbes that we saw on the early Earth. And so what we're going to talk about today are called biosignatures. And a biosignature is generally any type of data that we can look at and say there must have been life here. And so there's many different types of biosignatures, but today I want to focus on 3. We're going to talk about biomarkers, stromatolites, and microfossils. We're going to start with biomarkers. A biomarker can generally be classified as some chemical evidence that was produced by an organism, some organic molecule that we see on the Earth today in the rock record that helps point towards past life. And so to show this, I want to tell a story about my good friend, the sea sponge. We see that on the left. The sea sponge is an organism that we see across the Earth today, and it can produce a chemical called 24-isopropylcholesterol. There won't be a quiz, so don't worry too much about the name. So we're just going to refer to it as 24-IPC. When these sponges throughout Earth's history died, they would sink down to the bottom of the body of water in which they lived, and they would get buried by sand and sediment. And over millions of years, this sand and sediment would build up and eventually be compressed into a sedimentary rock. This burial process is called diagenesis, and this is how rocks are formed. Of course, the sponge itself has no hard parts, no bones, nothing that's able to be fossilized, and the sponge decomposes. However, some of the chemicals produced by the sponge, for example, this 24-IPC molecule, ends up being preserved. And we know that because 24-isopropylcholesterol is a type of molecule called a sterol. And when sterols are subject to the temperatures and pressures that occur during diagenesis in this rock-forming process, they transform in very predictable ways. Some electrons get shuffled around, some side chains fall off, and we end up with what's called a sterate. So when we look in rocks from time periods where we know that there were ancient sea sponges, we see evidence of these sterates. But 24-IPC is a very special molecule because when we look at the genetics for life across the phylogenetic tree, and we look at the building blocks that are necessary to build this biological molecule, we see that demosponges, this type of sea sponge, is the only organism anywhere in Earth's history that can make a significant quantity of this molecule. And so when this specific sterol decomposes into a sterane, and we find the sterane associated with 24-IPC, it gives us diagnostic proof that there was this specific organism at some point in the past. And this allows us to learn a bunch of very exciting things. For example, we know from just this biomarker evidence that sea sponges were likely the very first animal to ever evolve. We have no fossil, we have no record of their existence, but just looking at this geochemical analysis and understanding the history of these molecules, we can interpret the past environment that these sponges may have lived in and know that these sponges once lived on the Earth far before any other animals that we have evidence for. The next type of biosignature I want to talk about is called a stromatolite. So here on the left, we have a microbial community on the Earth today. This is from Shark Bay in Australia, and it's one of the only places on the Earth today where we have the active creation of stromatolites. What is a stromatolite? Well, a stromatolite is what happens when these microbial communities that live in shallow marine environments get covered by a layer of sand. But unlike the sponge that was dead, these organisms are very much alive and they're photosynthesizing. And so they want to travel up through this layer of sand to get into the sunlight. In doing so, they trap the sand underneath them. This process repeats, another wave washes over, sand washes over them again, and these microbes crawl their way to the surface once more. Over time, when this structure gets preserved in the rock record, you get something that looks like the image at the right. So you've got this very clearly banded structure that shows the growth of the microbial community over time. And so when we see a structure like this in the geologic record, we know with absolute certainty that there must have been life there, because you don't get this sort of shape otherwise. And so when we find stromatolites, it can tell us about the organisms that produce them, but also the type of environment in which they lived. For example, was the water fast moving or slow moving? What depth of water level was this? Was this salt water? Was this fresh water? We can learn all of that about the organisms and their environment just by looking at this geologic record of their preservation. Keep in mind, this isn't a fossil. There are no individual microbes preserved. They couldn't have been. They had nothing capable of being fossilized. But we do have this fossil evidence of their community, the structure of their community, and that can tell us a lot about their environment, both here on Earth, and we'll talk about Mars in a moment. And then last, we have microfossils. And I've been saying that there are no fossils of these organisms, and that's mostly true. Here we see cyanobacteria, and these are the bacteria that we think originated photosynthesis, so they changed the Earth in many important ways. But what we find throughout the rock record is evidence of these organisms being preserved, not in a fossil, but almost as a trace of where they once lived. So on the picture on the right, we see this white mineral, that's a carbonate, which we expect for these sort of marine environments. But then this brown mineral is a tube encasing one individual cyanobacteria. So this is a microscope section. And this is a silica rock, not carbonate. And so we know that there is some role that silica is playing in the preservation of these microbes. And this tells us a couple of things. It tells us about the microbe itself. We can see the shape and structure and morphology of one individual organism, which is exciting because it's the only record of these organisms from billions of years ago that we have. But it also tells us something about their preservation. The majority of life on Earth has never been preserved, and we never have evidence of it. But for some reason, these specific microbes in these specific communities were preserved. And we know that they're preserved due to interaction between minerals in the water, such as silica, with the rest of the depositional environment. This helps paint a picture for where we might find microfossils and where we find this evidence for life, because we know that preservation happens in these environments, in these shallow tidal environments, whereas in the deep sea, maybe this preservation wouldn't occur. And so this is all important because when we look for Mars, we're using these sources of evidence, these biosignatures on Earth, to help drive our search for environments on Mars that may contain this evidence for life that we're looking for. So when you hear scientists talk about searching for evidence of life on Mars, this is the type of evidence they're looking for. So here we see a picture of Jezero crater on Mars. And Jezero crater may contain all three of these types of biosignatures. Let me repeat that. Jezero crater may contain all three of these sorts of biosignatures. We have no idea if it actually does. We have to go and find out, but we know that it's at least possible, which is why NASA just spent almost $3 billion sending the Perseverance rover to this crater. And if we look at this crater from a geologic perspective, it makes a lot of sense why it was selected. For one, we see this inlet river. So it's a very clear river leading into this crater. And if we blow up this section of the crater and put it in false color, we can see this really beautiful river delta. And we see that we have rocks deposited here that only occur in the presence of water. So we know that this is unambiguous proof for flowing water on Mars. But if that wasn't enough, Jezero crater also gives us an outlet river that we see here on the other side. And this helps paint a picture for what Jezero crater may have looked like in Mars's past. We see water flowing in, being deposited, and then sitting in a slow-moving lake before being taken through an outlet. And so when we look at this crater, we see all of the elements that are necessary for the preservation of life. When we look at the deep past on Earth and see the sorts of organisms that were able to be preserved on the ancient Earth, a lot of those conditions are the same ones that we see in the Jezero crater. And so this is why we select this crater as the location where we're going to send our rovers, because it gives us the best opportunity to find this source of life, this evidence of life, if it exists on Mars. There's just one problem. Rovers have been a phenomenal robotic architecture for a very long time. They're scalable. We had everything from the Sojourner, the size of a small remote control car, to Perseverance, the size of an actual car, and everything in between. They're also incredibly energy efficient. They're stable. So if you lose communication with your rover, chances are when you reconnect with it, it's going to be right where you left it. And so there's a lot of benefit to using this robotic architecture. You can fit many different scientific experiments on it. And so they're a really, really great scientific platform. The one issue is Mars looks like this. We see steep slopes, we see slippery, sandy regoliths, we see large, jagged boulders. And the wheels of a rover are just not capable of traversing this sort of environment. And for the type of exploratory missions that we've done thus far, that's been fine. Rovers have and will continue to be incredibly useful at exploring that terrain. But in order to find the sort of geologic evidence we need to understand the past of Mars and if it had life on it, we want to find geologic outcrops that cut through Martian geologic time. And these sorts of outcrops don't just sit out in the middle of a field. They exist in these more extreme, tough terrains. And this is important because in order to get this data, we have to collect samples from Mars. We don't have evidence of these rocks on Earth. The only examples we have are small Martian meteorites like I have here. It doesn't taste like much. And if you did a biomarker analysis, you wouldn't see much on it either. So we have to go collect these samples in these hard to find environments. And so what I'd like to convince you of is that that future is going to come with legs. That in order to sample these environments effectively, we need robots that are able to have active stability. As the ground moves underneath the robot, for example, trying to walk across a sand dune, these robots are able to respond to changes in their environment. They're also able to run and jump and pronk and tiptoe around these difficult terrains in a way that rovers couldn't do. However, these platforms also have a lot of growth. They're still very young in their technological maturity. Many challenges remain. For example, long-duration autonomy is a big problem. We don't have legged robots able to go on these long-term missions. Stability is also a big issue. They require this active stability, which is a good thing for getting around the terrain, but it also means that they can fall over fairly easily. And for multi-billion-dollar missions, that's not something that we want to put our trust in. They also have some energy efficiency issues and scalability issues. They don't get very large. But every year, we cross more and more of these challenges off. And so the argument that I want to make is over time, especially in the next 10 to 15 years, and especially as we go beyond Mars, to explore environments like Europa and Enceladus, other planetary bodies that we believe may have life on them, this sort of mission, and especially fleets of legged robots, are going to become more and more useful because of their ability to locomote in environments that are too difficult and too unstructured for traditional rover architectures to traverse. I have no idea if there's life on Mars. I have no idea if there's life anywhere in the universe. But when I look around me and I see how astronomically huge the universe is and how astronomically huge the diversity of life on Earth is, I have to believe life is out there. So thank you for joining me on this little adventure, and I hope that you'll join me as we continue to explore our solar system and beyond. I have no idea what we'll find, but maybe that's the point. Thanks.
Thank you so much, Kyle, for that inspiring and really interesting presentation about microbes, minerals, and search for life on Mars. It's really inspiring. Thank you so much. Okay, I'd like to now go into the question and answer section. I see a lot of questions coming in from the chat box. I'd really like to thank you guys. As you know, Dr. Sharmila is not here. We'll try to answer as much as possible, but I have Kyle here, who is going to definitely help us answer some questions. So I'd like to start answering them. And if you have any more, you can keep shooting in the chat, and we'll try to answer all of them within the time. So, Zoom. Okay, the first question was from Luca, and it was, are there any applications that astronauts are using today that came from experiments on animals on ISS? So I don't know if, Kyle, do you have any examples? Maybe you're not exactly the specialist on this field, but if you have anything.
Yeah, I know that that is occurring, but I couldn't point you towards any specific examples, unfortunately. I work with NASA JPL, which is primarily robotic exploration, so human spaceflight is a little out of my wheelhouse.
No worries, thank you. Luca, please make sure to write it in the questionnaire form so that Sharmila can see as well. I've seen that a lot of the rodent research is used for, is really a test bed for the astronauts. So the applications are I think there's a bunch of different applications that are helping the astronauts' lives become much better for them. I'm sorry, I don't have any good examples. If there are any people in the crowd that knows anything, please shoot them in the chat. You are all welcome to put in your comments there, and I see a lot, so that's great. But yeah, definitely make sure to send the question out in the questionnaire form. Okay, the second question was from Madhavan, which was asking about the cold atom lab, if it's being used for food preservation for manned missions on the moon and long-time ISS. As I wrote in the chat, I'm not really sure. From what I saw from the website, it doesn't look like they're doing anything for food preservation at the moment. But yeah, if you can write that in the questionnaire form as well, that would be wonderful. The next question was from Carlos. Are there any changes in synapse under microgravity? Actually, we had a lot of talks about things more biology and physiology in the morning session. So we had professionals from the Belgian Nuclear Center and from different places talking about T cells, tissue engineering, wound healing, and everything. So maybe if you can check the morning session that I will put up later in the week, that could help you as well. Do you have anything to add on this, Kyle? For
not particularly.
Yeah. If there's anything, if you guys know anything in the chat, I'll ask you guys to help out with me on this. Another question from Carlos was, is there a center or laboratory working on nanomaterials-based therapy against brain damage in microgravity? This, we will have a webinar next week on physiology. One of our professionals is focused on brain cells, and I'm not sure if it's brain damage, but she's focusing a lot on the effects to the brain. So if you can join us next week, I think you will get more information as well. Another question I saw was from Madhavan, which is to Kyle, which is asking about the robotics, if it's a media lab or a CSAIL. I'm not really sure what this means for me. Maybe Kyle, you can elaborate on this.
Yeah, those are two different labs at MIT. When I was at MIT, I was in the aerospace engineering department and the geology department, but most of my research through MIT was through the geology department. And so I was working with Tanya Bosak and Roger Summons, and Tanya Bosak is actually one of the science leads for the Mars Perseverance mission. Now for robotics, I work at UT Austin in the robotics portfolio program with Dr. Louis Sentis. So I did a little bit of work with CSAIL, but that was never the core partner in my research.
Perfect. I think that was a very clear answer. The next question I see was from Annie. She was asking, may I know the names of the craters where the biomarkers on Mars were mentioned?
So this was Jezero Crater, and we have not found biomarkers yet. I want to make that clear. It's exciting because it has the potential to contain these exciting biomarkers and biosignatures. It's where we currently have the Perseverance rover right now on Mars. So that's Jezero Crater. I can spell it in chat, but there's no biomarker that's been found yet. Once we do, you will see it. Well, if we do, you will see it all over the news. It will be the first proof of life outside Earth ever. And we're hoping that we find it, but we'll see.
Okay, perfect. Another question, I think this is for you, Kyle, as well, from Vivan Raji. Are there open source space robotics projects that the general public can contribute to? So if you know any.
Yeah, not specific to space robotics, but for robotics, absolutely. And as a field, it's all growing very, very rapidly. So as the field of robotics grows, space robotics pulls research from the robotics community and tests it for the space environment. So there's tons of different robotic and algorithmic open source projects that you can work on, different AI programs. I'm not sure if I have any great ones off the top of my head. It's such a large field. There's lots of different projects to work on, but any open source robotics work is going to end up trickling down and helping space robotics research, because that's the way NASA works. Is we're not taking the most cutting edge research. We have to prove and test and verify that this work works. So the more open source work that we get from the robotics community as a whole, the bigger the wealth of information we have to pull from when we go to build these robotic architectures to send to other planets. So any open source work is hugely beneficial to the NASA and space research community in general.
Great, thank you. Another question to you is, what is the relationship between the evidence of water and biomarkers and markers?
Yeah, and Mars, I think they meant. Yeah, this is a phenomenal question. So evidence for water we already have found on Mars. And so the way we have to think about this is these biomarkers and other biosignatures we find in rocks that only form in the presence of water. So none of these are coming from igneous rocks or metamorphic rocks. These are coming from sedimentary rocks. And sedimentary rocks are forming by layers of sand and silt and deposit that is being deposited from water systems that then build up over time and are compressed into a rock. So when we look for a biomarker, or we look for a stromatolite, or we look for a microfossil, for one, these microbial communities that form stromatolites and form these microfossils, we know that all of them live in water. These are not land-based organisms. And so when we find these in a rock, we know immediately that there must have been water because these rocks couldn't have formed any other way. And so the biomarker itself isn't necessarily saying because we found a sterane, that means that there was water here. It's more of we found a sterane in a rock that only forms in water, therefore there must have been water here. And so we already have done the first part. We know there was water here. Now we're trying to find the steranes and other molecules like that. For example, hopanes come from hopanoids, and pigments do the same thing. And I see someone else over here that the software for the Ingenuity Mars helicopter is open source, and that is correct. So that is one great way to get involved.
Okay, perfect. Thank you so much, Kyle. I think we've gone through all of the questions. If you have any more, this is your last call. But thanks, Kyle. You've helped a lot in answering all the questions, so thank you so much.
Yeah, thank you so much for having me.
So I think we can call it a day. So before you guys leave, please make sure to answer the questionnaire form. Your feedback is really important to us. We'd like to provide better webinars for you. So really, we'd really like to hear your feedback. Also, next week, same time, same place, we will have our lecture on physiology. As I've explained earlier, we have two sessions, one in 10:30 Central Eastern Time. We have one at the same time, so 4:30 Central Eastern Time. We have different speakers for both sessions. We have different professional speakers and different student speakers, so make sure to tune in. Wenbin, my colleague Wenbin put up a registration link, but if you've already registered for this one, so the registration is for webinars 2 to 9. So if you've already registered, you don't have to register again. We'll send you an email again on Tuesday, the day before the webinar, with the links. And this is being recorded. All the presentations can be found on our website. So if any of your colleagues or friends or family missed out, please direct them to the website so that they can see the recordings and the presentations. Okay, I really want to thank Dr. Sharmila. She's not here, but thank you so much for the amazing presentation. And thank you, Kyle, for this inspirational presentation. I think it was really interesting to everyone. I think you guys all had a really nice overview of biology. So as I told you, next week, we'll dive into physiology. So I really look forward to having you all participate next week as well. So thank you so much. Thank you for your engagement. And yeah, have a nice day, everyone. Bye. Take care.